right, when we talk about the plasma membrane, we said that the plasma membrane is selectively permeable. Meaning it allows for some substances to cross or move into and out of the cell. Now, before you can understand transport, there's another definition that you need to understand called a concentration gradient. But before you talk about a concentration gradient, you need to understand intracellular fluid and extracellular fluid. Now, we're going to go over term concentration gradients. Excuse me. Okay, now, molecules are going to be suspended in the intracellular and extracellular fluid. Molecules tend to have a difference in concentration on the inside compared to the outside. So basically, we're looking at the concentration or the amount of substances that are dissolved in the intracellular fluid versus the extracellular fluid. This is called a concentration gradient. Alright, so I'm going to show y'all some concentration gradients differences. When you look at the outside of the cell, you can draw a lot of dots to represent a number of molecules that's dissolved inside of the extracellular fluid. So molecules are dissolved in the fluid. So, the intracellular fluid has its own amount of molecules. So, when we look at this, we would say that there is a big difference. There is a very high concentration on the outside. This is a very small concentration on the inside. So we compare the number of molecules on the outside in the extracellular fluid versus the number of molecules on the inside of the cell. That is what we call a concentration gradient. Now it can be reverse. We would literally call this a high concentration because there's more molecules. This would be considered a low concentration gradient. Low concentration on the inside compared to the high concentration on the outside. You can flip these. You could put a lot of molecules on the inside and you would say there's a high concentration on the inside. there's a low concentration on the outside. These can be changed, but before you can understand the two different types of transport, you need to understand this definition of a concentration gradient. Now, there are two types of transport. Across the plasma membrane. Now, basically, when you do the two different types of transport, there's these long definitions. Passive transport, passive means no energy. This is going to be the movement of molecules from high concentration to low concentration. When you use the word passive, it does not require energy, which is ATP. That's what the word passive means. So literally, this is going to be the movement of molecules from a high concentration to a low concentration. It's going from high concentration to an area of low concentration. That's all passive transport is. So the next type of transport is active transport. This is going to be the movement of molecules from its reverse, low concentration to high concentration. The word active, think ATP, it requires energy, which is ATP, the energy currency of all cells. That's what active transport means. So it's literally the same picture, the plasma membrane, but now you're moving your molecules This is a high concentration, this is a low concentration, from low to high. So you're moving substances out. Now, there's other definitions that kind of go with these, or there's different types of transport, but there's one way that I teach transport in my classes, and I literally think this is the easiest way to think about it. When talking about transports, I always think of like going to a concert. Like whoever you like. If you like Lizzo, if you like the Rolling Stones, you know, they still are performing. I actually had concert tickets and they got canceled. Anyone you would like to see, literally concert, in live concert. Now, everybody and their dog, when you go to a concert, wants to be at the very front. Literally, this is what we call a high concentration. If you've ever been to a concert, everybody wants to be at the front. It is so packed with people. And as you move back to the back, you know, you have groups of people, you know, that are together, but it's not as packed as it is at the front. Now, I use this to help my students understand the different types of transport. So, this obviously represents a high concentration. There's more people at the front of the concert than there is at the back. There's a lower concentration in the back. Now, you can make this super easy on yourself by learning this way of drawing. Especially when you're on a test and you're trying to think about it really quick. When you move something from high concentration to low concentration, this is called passive transport. Now, there's another thing that they include in the definition of a passive transport, which I have not taught you. When you say passive transport, they literally will say it moves down the concentration gradient. Down the concentration gradient means it goes from high to low. That's literally what down the concentration gradient means. So, I like this analogy of the concert because my arrow is going from the front to the back. If you did the arrows like this, that would be going down. This would be up and this is down. So it's going down the concentration gradient. That's what this arrow represents. It's going down the concentration gradient, which means it's going from high to low. Now, it's passive. If you are at the very front of the concert and you're like, say, up front and center, say that literally this is you, you're right here, and you want to leave, everybody is gonna split. They're just gonna say, yeah, I'll take your spot. So it's really easy. It does not take energy, but say this is your favorite band and you want to go to the very front and center. It takes a lot of energy to get from the back all the way to the front and center. People are gonna put their elbows up, they're gonna block you. They're literally not trying to let you go to the front of the concert. I've seen people like rope off their chairs. So if you want to go from the back, from a low concentration of people to the front and center, now you're going up. So this is against the concentration gradient. You're going against all these people. They're not going to let you through so easily. It's also going up, but more it's going against people. They're going to band together. They are not trying to let you go. This is called active transport. Now, when you use the word active, you have to use energy to get to the front. This does require ATP. It requires energy. It is active. You have to be active to go against everybody to get to the front of that concert. So this is literally the easiest way to remember passive and active transports. Now, you're going to have several subtypes which fall under each. When we talk about passive transport, there's several types. that I'm going to discuss. Simple diffusion, facilitated diffusion, and osmosis. Osmosis literally has its own video. For active transport, we're going to talk about the sodium-potassium pump. Now, sodium is Na, K is potassium. This is how you write out the sodium-potassium pump. Then you have the use of vesicles, which are like little carriers. So if you use vesicles, Vesicle Transport, there's going to be two. There's endocytosis. Endo means in to the cell, cyte cell, which is going to have several types as well. That's going to have Phagocytosis, which means to eat something solid. Let's think of esophagus. Then you're going to have Pinocytosis. Pino, I always think of pina coladas, which is a drink that's bringing into the cell like liquids. And then receptor mediated endocytosis. And then the second thing, and I'm running out of paper, so I'm just going to raise this up and use a new sheet. Using vesicles is exo for exit, cyte for cell, tosis. So these are active transport, but all active transports goes from low concentration to high concentration. These are just subtypes that go with it. So if I ask which type of active transport, it's one of these. Which type of passive transport, it's one of these. So the first thing that we're going to do is we're going to cover passive transport. And the way we do transport, and when we're talking about transport, it's transport across the plasma membrane. So, first thing you do when you do transport across the plasma membrane is you draw the phospholipid bilayer, a fluid mosaic model. Now, I'm only going to draw one set of proteins, integral proteins, but its classification or its function is transport. Now, in class, when I think of the phospholipid bilayer, I literally discuss how this literally looks like a chain link fence, a double chain link fence. So, if we were talking about molecules that are very small, like oxygen gas, anything that's small. Now, this could be anything that's small. You have more abundant things on the outside than you do on the inside. So when I discuss this, and I say this is like a double chain link fence, think of this as a prison. You're selectively permeable. So if in your extracellular fluid you have a high concentration of stuff, and on the intracellular fluid, which is the inside, you don't have that much stuff. Think of this as a prison. You're going to have more of everything on the outside than you do on the inside of a prison. So the first type of transport that we discuss is the different types of passive transport. The definition of passive transport is the movement of a substance from high concentration to an area of low concentration going down the concentration gradient. So our first type of passive transport is simple diffusion. When you say simple diffusion, which is what I'm drawing here, so this is passive transport. So we're going to move substances from high to low, but this is going to be what we call simple diffusion. Simple diffusion is literally the movement of small, very small molecules from high concentration to an area of low concentration. So when you think of simple diffusion, this is going to be like oxygen gas, CO2, anything that's very, very small molecules. Now, back to my analogy of this is a prison. This could be cigarettes, it could be little bottles of alcohol, very small things. If you pulled up on a prison and you want it to pass very small molecules through a double chain link fence, it's possible because there's little tiny gaps. So, for simple diffusion, only for simple diffusion, you're gonna take these small molecules, and it's simple, they're gonna go through the phospholipid bilayer. So it goes from high concentration, you move it, molecules from high concentration to low concentration, down the concentration gradient. This literally is going to go through the phospholipid bilayer. Because it's got gaps in it. That is simple diffusion. Very small molecules go through the phospholipid bilayer. Now, when we say facilitated diffusion, I'm trying to draw this very simply to make this a lot easier. If this is a prison, the proteins that we say or we see here are called transport proteins. We call them transport channels. Now, the difference between simple diffusion and the next one is called facilitated diffusion. Facilitated diffusion is going to be the movement of very large molecules from high concentration to low concentration. So I'm going to draw some over here because I kind of ran out of room. It's okay. We're going to draw another transport protein. You know, a prison has more than one gate that you can enter, right? More than one entrance. Now, we're going to be talking about, for facilitated diffusion, very large molecules. C6H12O6. That's the molecular formula for this molecule, which is glucose. So these are very large molecules. Now, very large molecules are kind of like, if we're saying this is a prison, you know, the iPhone 6 Plus or 8 Plus, anything that's super large, fifth of vodka, anything that's large, TVs, cell phones, you know, things that you shouldn't have in the prison. These are large molecules. When you say facilitated diffusion, you're now taking the word large molecules and moving it from high concentration to low concentration. So you can say that these are cell phones. You still are saying it's a passive transport. It's the movement from high concentration to an area of low concentration. So you're moving it from high concentration on the outside to a low concentration on the inside. However, one key difference is that it's not simple. It has to have a helper. So when you use the word facilitated diffusion, it's large molecules. However, these are gonna go through protein channels. They're too large to pass through the phospholipid bilayer. So the only difference between simple diffusion and facilitated diffusion is the method through which they pass. Now, osmosis is the third type. Osmosis is going to be the movement of water, which is the universal solvent, from high concentration to low concentration to the area of highest concentration of solutes. A video is posted on osmosis. Watch it. There's a lot on osmosis. All right, so that's the different types of passive transport. Now, active transport. It's active, energy required. If you're using the concert definition, movement from a low concentration to a high concentration against the concentration gradient, ATP or energy is required. Now, the first one that we talk about is the sodium-potassium pump. This is one of the most important of all of your transports. Now, normally, you have a high concentration of sodium on the outside of the cell. This is where sodium is. And normally on the inside of the cell, this is where potassium is. You have a high amount of potassium on the inside and a low amount of potassium on the outside. Now, the sodium-potassium pump is going to move sodium out of the cell, and its job is to move potassium into the cell. Remember, you're going from a high concentration to an area of low concentration. So its job is to basically restore the cell back to normal conditions. So the way you do this is when you have sodium on the inside, you're going to pump it out. So you pump out 3 sodium, and then you have potassium that you need to pump in. And that's the sodium-potassium pump. You're trying to restore it back to its normal conditions. So it's going from low concentration to high concentration. All right, now the next thing is vesicles. Vesicle transport. Vesicles are little tiny sacs that carry substances inside of the cell. So if we were looking at the cell in the lab, there's literally like these little sacs. A vesicle is a little membrane-bound sac, and inside of the vesicle, it's going to carry substances. A vesicle is literally a little sac inside of the cell that transports substances across the cell. So this is a vesicle. It is a membrane-bound sac, which means it has a membrane in it. It carries or transports substances. Now, a vesicle can release its substance out. So if this travels to the plasma membrane and it releases it, this is exocytosis. So the substances are going to get released. Now, if you have substances that are coming into the cell, a vesicle will pick it up This is called endocytosis. So, we're going to talk about vesicle transport. The first type we're going to talk about is endocytosis. Now, the molecules that you bring in, if they're solid, so this is food, that would be phagocytosis. It's going into the cell. Phagocytosis literally means the cell is eating, to eat. So that's literally what phagocytosis means. So this is bringing solid substances into the cell. The next one, Pinocytosis. Think of pina colada. This literally means to drink. So if these molecules are fluids, liquid, or fluids, then you would say that this type of endocytosis is pinocytosis. Now, another type of endocytosis is if you had a receptor. Remember we said our glycolipids and our glycoprotein. Say you have a bacteria, a bad guy that we recognized as foreign, and you need to bring him into the cell to destroy him. You would take the receptor and the bad guy, and it would go into the cell. So this is endocytosis. This is called receptor mediated endocytosis. So you're using your glycolipids and your glycoproteins, which recognizes foreign bacteria, and bringing it into the cell. So there's 3 different types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. But vesicle transport exocytosis, the vesicle is going to release the substances or let the substances exit the cell. There is also another type of transport that involves vesicles, where it like maybe brings it in and then releases it on this side. So if it goes from one side to the other, it's called transmembrane cytosis. That literally means transfer it from into one cell and out the other side. That's transcytosis. So basically those are your different types of transport. I highly recommend watching the video on osmosis.